So let's look at transcription and translation, okay? We've copied it all up. We've seen how that does, replication forks, semi-conservative, blah, 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 blah. But now let's look at the nuts and bolts of this central dogma, okay? I want to copy this SOB in the nucleus. I want to get it out into the cytoplasm, and then I want to translate it. So we've got to make a copy. And that's what the first step is. It is transcription, all right? This is step number one, okay? DNA is serving as a template, and I am scribing it out. I am writing it out what it is. Shorthand in mRNA. Where does this occur? It occurs in the nucleus, okay? Transcription occurs within the nucleus. Ooh, excuse me. Okay. So we whip this thing open. We copy it. Just like we saw with the replication bubble, it's the same basic thing that's happening there, except we're going to use a slightly different enzyme. It's just RNA polymerase. That RNA polymerase is going to come in and really open up the gene, okay? And we're just doing a specific segment. You know, we have this entire strand of DNA, but we only want to code, I only want to transcribe what I've been told to transcribe. Okay. And this is all done through chemical messengers. All right. So the gene is going to unzip in a region where it's supposed to, all right, where it is supposed to happen. All right. So we're going to open it up in the gene area that I want to transcribe. All right. The gene codes for the protein. So we're going to open that up and we're going to copy, and we're going to keep going. We're going to make this copy of this gene until we get to a region where we're told to stop. There's going to be a start, there's going to be a stop, right? We talked about this with the genetic code. Well, we're going to continue for as long as we need to until we're told to stop when we're transcribing, all right? That entire gene that gets transcribed, that gets written down, okay, is referred to as an mRNA transcript, okay? A transcript is just a written down thing of what happened. That's all we're doing here, okay? So I've taken my DNA polymerase, I've opened up that segment where the gene starts coding for it, and I start copying, and I go until I hit a stop message. This is referred to as the mRNA transcript. Now, this could be hundreds of genes, this could be thousands of genes. So the length in which this occurs, I'm not going to say, you know, it's only like 15 base pairs. It's really long, okay? There are thousands and thousands of genes within there, and there may be several genes that code for a single different thing. So we have to copy as much as we have to, all right? What do we copy? We obviously copy the one piece of the one side of the DNA molecule. And it's referred to as the sense strand because it is helping us to make sense for what I'm transcribing. And it helps to produce the sentence for which I read to make the protein, all right? So the information of the specific gene, the base sequence, okay, is on one side of the DNA strand, all right? So when I unzip that DNA, okay, I only have to copy one side of it, all right? This gives me a general sense of what I need to do, okay? And here it is, okay? Here's my RNA polymerase, okay? That's what helps to open up this DNA molecule so I can get in there and make my copy, my messenger. My messenger carries the written word. My messenger carries the postal stamp, okay? That package, right? The letter. So the mRNA molecule carries the message, the letter, out of the nucleus into the cytoplasm for the second part of this whole central dogma of biology. The genetic information in that gene has been transcribed, just rewritten, okay, onto that RNA molecule. Okay, and this has all of my sequence that I need to be able to read to piece together the proteins to make, or piece together the amino acids to make the protein that I'm ultimately looking to get. Here, and this just shows you, here's my sense strand, the template strand, okay? And there's my RNA polymerase unzipping a region and making magic happen.
During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other.
Okay. So I have opened it up. I have copied my gene, the gene that codes for a protein. So now, is it done? We wish. We have a few more steps that we got to do before that little piece of copied genetic information gets out into the cytoplasm to be translated. So this, what's called pre-mRNA, okay, will need to be processed prior to leaving. Okay, we just kind of saw that there. Excuse me, in that video. So we got to make a few different modifications. The first set of modifications to get this mRNA to be mRNA is we have to put a cap and a tail in it. Okay, we have to have a cap and we have to have a tail coat. All right, just think of it. In olden times, people dressed up to go out and about by putting on a cap. And we had to put on our tail, our tail coat. RNA polymerase. Open this segment up. Copied it. Copied as long as we had to. Could be a few hundred, could be several thousands. Okay. Of amino acids long. We've copied it. Now I put a cap and my tail coat on. The cap ensures that translation out in the cytoplasm begins where it's supposed to. The poly A tail protects it as it's heading out into the world. Okay, so it doesn't get broken down by any enzymatic action. Facilitate its movement out of the nucleus. The last thing we have to do is we have to cut and paste. What do we have to cut and paste? We have a couple different segments in all pre-mRNA. We have these things called exons, and we have these things called introns. Exons are actually the good stuff, okay? The exons are what we are going to read out in the cytoplasm. These are what are going to be ultimately expressed, and by expressed, we mean turned into proteins, which will make structure, and structure dictates function. Introns, I'm going to put this. They are junk DNA. And we put junk DNA because they don't really code for anything. We honestly don't know what the hell they do. We have a few good ideas and we have a few good working theories in it, but we are not exactly sure 100% what they do, which is perfectly fine. We know what they can potentially do in a few different areas of research, but we don't honestly know 100%. Okay. So we need to splice, we need to cut out those introns. So to do that, we call it RNA splicing, okay, where we splice out the introns. To do that, we are going to need, shockingly, another enzyme, okay? And this is what it's called, okay? It's just a spliceosome. It's an enzymatic body that goes in and it'll attach on one end, another one will attach on another end, and snip. And when it snips, it glues the two exon segments of the exon together. Once we do that, ta-da! Now we have our mature RNA transcript. Holy smokes. We started this nonsense way in the hell back here, right? Where we opened it up, okay? Where we're going to make a transcript of our mRNA, right? We unzipped it. We exposed that segment of the gene. We copy that gene, okay, through that RNA polymerase. And we make this thing called a pre-mRNA transcript. We have to modify it, okay? It's almost like sperm development, all right? Sperm isn't fully mature until you cut away all the cytoplasm around the tail and the mid piece, and you put a little acrosome cap on him. The same thing here, okay? We have to put a cap on. The cap ensures that the ribosome attaches here. Hey, right here, this is where you're supposed to attach and start reading from this point. We have to put on a tail, and the tail ensures that it gets out of the nucleus, and it also gets, it survives the enzymatic action, the enzymes that are floating around within it. Also, as we put on our cap and our tail, we have to cut out the bad. And by bad, I mean the stuff that doesn't code for anything, and that's what introns are. Introns are segments, very long segments, of junk DNA that does not code for anything now. Did it? Potentially. Does it do some other potential stuff? Yeah, and we'll hint at that here in a second. But right now, as our DNA stands, it does not code for anything. So we got to splice it out. So we have spliceosomes that get in there and snip and glue it back together. Okay, snip out the introns, glue together the exons. Now we have our mature mRNA transcript. In prokaryotes, they have the same jazz, but they have their introns will self-splice, okay? I honestly don't know the significance of that, except the simplicity of it. If somebody wants to look it up and let me know, I would love to know, okay? But I got too much crap going on in my life. So if someone else figure it out for me and let me know. So here's our DNA. Got codes and no codes, codes and no codes. Well, RNA polymerase, open it up, unzip, and copy. And we get a copy. We put our cap and our tail on, and we splice out our introns, and ta-da! Now it can go out into the world and do some stuff.
The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. The strand.
So that's where we're at here. Get my pen back. Introns, they can potentially do some things. It actually gives us more genetic information for which to pull upon, because even though that segment doesn't code for something, it actually can, if we do have a misread, and let's say we have a segment of a nucleotide that gets deleted, then maybe that next letter, that first letter of the intron, it will move up and now it'll be translated. And maybe it may actually give an entire different protein, which may be okay. That protein can maybe give somebody an advantage. Okay, give that individual an advantage. And that is just what evolutionary change is, just a random act of mutation. Okay. So this opens up a kind of whole new world in how our genome is handled or is maintained, because we have a number of regions. The vast majority of our DNA doesn't actually code for anything right now. And it takes millennia to get this stuff to adapt and change. And we'll talk about that when we talk about evolution. Okay. But the more complex the organism, the more exons and introns are going to be available. And the more introns are there, the more varied a potential genome can be. So, it can give a multitude of combinations. Okay. They actually have been shown to help regulate gene expression, turn genes on, turn them off, turn on or off. Okay. And I'm trying to figure out a way, to get this video to show. I may have to make a recording of a recording, so I don't know how the hell I'm going to do it. But where this is what they figured out with certain segments of chromosomal segments, is there's a segment that actually is basically a switch, and it's these introns, and it doesn't code for anything, but it codes for a switch, and that actually can potentially turn on a genome segment, which can give rise to arms or to fins. In the example in that little movie, in that documentary. It may encourage crossing over during meiosis. And remember that crossing over in meiosis gave a buttload plethora, if you will, of various different genetic outcomes. And variation is what drives sexual reproduction. Okay. All your brothers and sisters may look alike, but they're not exactly alike. It's just that little bit of variation that exists. That's what sexual reproduction allows. And they show an example of kind of moving genes in between species here. And this is what we're going to talk about in chapter 14 in the next lecture, where we talk about the ability for us to understand where genes specifically are, because we've made a genetic map. We've mapped the genome of many different types of species. So being able to kind of know, I can either tag a molecular sequence, and I can do a number of things with it. I can perpetuate that little segment. I can grow that segment. I can enhance the ability to turn on or off that segment in that species, or I could take it and put it into another species. And that's, I made reference to that earlier with the glowingness of the jellyfish here, and you can insert it in multiple different types of organisms, and you can still all get the same response, because that's what that gene codes for, a fluorescent protein production in the species. And we'll talk a little bit more of that biotechnology application of the understanding of this central dogma here.